Well, greetings. It's kind of fun to be here. I was raised in the Midwest, so this feels like home, okay? So it's like, yes, rain, cornfields, those sorts of things happening. It's like, yes, this is home. Okay. Well, this evening here we're looking at the case for intelligent design. And I'm going to give you a little bit of background, just sort of like Alan did with his things, so that okay, what do you mean by intelligent design and that sort of a thing. Definition here is fairly important. It's basically this. Look, the theory of intelligent design simply says that certain features of the universe and of living things are best explained by an intelligent cause, not an undirected process such as natural selection. So again, it's a very, very limited claim here that's being made in the context of intelligent design, that science can provide evidence that there's a designing intelligence behind things. And now some people go, whoa, gee, there's like religious implications to a theory like that. But a lot of scientific theories have theological implications. The Big Bang Theory does. Like, who started the Big Bang? Darwin does, right? How did life come to be? How did life change in its various formats? So it's not unusual for a scientific theory to have theological implications, and intelligent design is one of those. Now, what it does not try to address is who the designer is. You'll see me venture off of that tact a little bit here as a Christian, but the basic idea with intelligent design or mere design is that you can recognize that an object is designed, but not know, who made that pot, or how old were they, or would I really like to know them? That kind of a question. And why did they make it? Was it a joke or a wedding present or something like that? And I don't have to know anything about making pottery or having taken classes to recognize that that is a designed object. Another important aspect here, too, is that it doesn't have to be a beneficial or optimally designed object, right? Because bombs are designed. So it's just a mere, okay, that is a designed object, is the recognition for it. And it's not, intelligent design itself is distinct from creationism, where you generally start with, say, Genesis in the Bible, and then say, okay, how do we correlate things we see in nature with the Genesis account? This is just stepping back and saying, no, wait. Can we recognize that things are designed? Mere design, again, is the idea here. So William Dembski has written on this. Intelligent Design is one of his classic books. And the way he argues it is that basically intelligent design suggests that we should add a third causal explanation to the way that science normally works. And this is a diagram that he has where it's like, okay, normal science works by looking for things that happen due to laws, right? Physical laws, chemical laws, that sort of things. These are things that happen of necessity. And then there's things that happen by chance. If you throw a bunch of Scrabble pieces on the floor, you get this arrangement of letters on the ground. And you could throw Scrabble pieces on the ground and pick them up and throw them and pick them up and throw them. And any particular pattern that you see on the ground happened by chance and is basically random. You'd never see that same pattern again if you kept throwing 100 Scrabble pieces all the time. So there's many events in the world that we can describe as happening by chance, just randomly. But you see design when you get both a very complex event, like what was that precise order of those random Scrabble pieces on the ground, with some specificity to it. In other words, good grief, those Scrabble pieces spelled out to be or not to be, or something like that, some very significant amount of English letters or something. So that would be the sense for this, is specified complexity, not just complex, not just specified, but this combination of the two. Now, if it's a human who's behind the design, okay, no one has any problems with that, unless you're a conspiracy theorist about Shakespeare or something. But the idea here is that we can look for things in the world around us and recognize that same sort of a pattern. And the major thing that you see it in is what we would refer to as information, specifically semantic information. A fancy word or a pair of words for, okay, there's a pattern here that does something independent of its own structure. For example, I'll get to DNA. So there's a code on the DNA that makes proteins, something completely independent of the DNA itself. That's the idea that we're looking at here. Again, Dembski would argue that information does not come from law, does not come from chance, and a way to illustrate it here with Shakespeare is to note that Shakespeare did not get his soliloquy for Hamlet to be or not to be by pouring ink on paper, blotting it off, and there it was, to be or not to be. That is, the laws of chemistry and the way ink interacts with paper did not give rise to those letters on the page. That required an intelligent mind shaping that ink on that paper to give rise to that particular thing. So that's what we're looking for, but we're looking for this in the what we might call the natural world. Well, people will bring up a problem with it. Basically say, no, you can't think about design in the universe, something like that. If you bring that in, it'll just kill science, because science historically has focused on finding natural causes for everything that happens out there, okay? And if we open it up, it's just going to let any weirdo in the door, So I just have to pause with that and say, wait a second. If you introduce design, as again, a possible third explanation, law, chance, design. If you introduce the possibility of design, does that stop science, or does that justify science? Now, if your head just did that, you don't know the history of science. So let me give you a background for that. One thing to mention first, much of what science does today, maybe even 95 to 98% of it, focuses just on those mechanical nuts and bolts for how things are interacting with each other, and design isn't necessary to evoke to explain why hydrogen and oxygen, mix, and you get a lot of heat, and you get water vapor and so on out of it. That, at least from a Christian perspective, God's providence and God's control allows material causes to be the explanation for the things that we see. So again, if you're a scientist in the audience is like, okay, 95, 98% of what's done in the sciences really isn't grounds for talking about intelligent design, but because we have mechanical causes that can explain things very, very well. But historically, it's interesting to see that the idea of design in the physical world, in nature, was actually a major assumption that God's science started. And if you look at the history of science, even Galileo, but you can add Kepler and Bacon and Boyle and Newton and Faraday and Maxwell, all these founders of science that we recognize today, they're all coming at it from a Christian background, interestingly enough. And that Christian background gave them the thought that, wait, the universe is created by a God who's established it to work in regular patterns. He's designed it to work in regular patterns. I wonder if we can figure those out. The world looks kind of messy, it looks a little bit chaotic, but maybe because we're made in his image, we have enough of a spark that we can see the design, the patterns that God put there. So actually the history of science begins with that expectation of patterns in nature, and that's just not taught in science courses today, unfortunately. That wait. Science started with a presumption of design, and that design ended up as Newton's discoveries of the law of gravity and Faraday's discoveries of the law of magnetism, Maxwell's discoveries of again, what became known as Maxwell's equations. All of that comes from a presumption that the universe is designed and that you can actually figure it out and maybe see the patterns that God put there. If you come at it from a secular perspective, you don't have any basis to presume that there are regularities there. So the assumption of regularities that got science started was because people were thinking in terms of design in the universe. So historically, it's interesting to see how that's been sort of forgotten. The universe was seen as intelligently designed as science is getting started, and it's been since forgotten. Well, again, how do you recognize design? This is part of the problem. And again, if you say that, hey, I think something here in the world is designed, well, there's disciplines that don't have any problem recognizing design. If you're an art critic, you don't look at the Mona Lisa and go, let's see now, how did that ink or how did that paint evolve and change over generations on that canvas to give rise to that smirk that she has, right? Or that weird background. I mean, where did that come from? How did that evolve? You don't ask that sort of a question. You recognize that it's designed, and then you ask a whole bunch of other really interesting questions, but not how did that, through numerous successive slight modifications, come to be the way it is. So again, recognizing design doesn't kill science. In fact, it got science started. And there's still plenty of scientific questions you can ask about things that you might conclude are designed. So again, it doesn't kill science, is the point. But while I'm talking about science and backgrounds, I should just mention this point. And if you fall asleep after this slide, you're okay. Because to me, this is the big point here. Don't fall asleep. I'll try to keep you awake. That's my goal. The biggest issue that I encounter in these sorts of discussions about science, intelligent design, and God's role in nature and so on is precisely how you define science, okay? And again, I think the way most people understand science is mistaken, sadly. Because most people would say, oh yeah, science seeks truth, right? What's really going on out there. We're trying to discover the way things really work and so on. But if you look at what science does, and you read the journal articles, and you read the reports in the paper and so on, what you find science always doing, although it's claiming that it's seeking truth, is that it's always offering the best naturalistic explanation for things. And after you watch this go on for a few years, and this took me a while to finally figure out, wait a second, it's claiming to be seeking truth, but all it's ever telling me is the best naturalistic explanation for something. There's a worldview that's packed into this, and the worldview is basically naturalism, that all there is in the world is natural things. There's no supernatural, there's nothing beyond the natural. And this is not talked about very much in the scientific world. It's just kind of assumed, if you will. And to give you one of the rare situations where this actually is discussed, Richard Lewontin was a geneticist at Harvard University at the time that he wrote this book review. And he's no longer with us, unfortunately, but he is one of the rare people who sits down and says it in fairly plain English. He says, look, we take this side of science, in spite of the patent absurdity of some of its constructs. He's talking about Darwinism and in spite of the tolerance of the scientific community for unsubstantiated just so stories, like how did the giraffe get its long neck? Well, it kept stretching to reach the leaves on the trees. Or how did the leopard get its spots? Those sorts of just so stories. Why? Because we have a prior commitment, a commitment to materialism or naturalism. And he goes on and says, look, let's see, click too many things here. There we go. Okay, it's not that the methods and institutions of science somehow compel us to accept a material explanation of the phenomenal world. So it's not that scientists have been looking for centuries for any evidence for God and just have not found anything. No. On the contrary, we're forced by our a priori adherence to material causes. A priori. Before you even walk into the lab, pick up your calculator or your pen, you're committed to finding material causes to explain everything. That creates an apparatus of investigation, a set of concepts to produce material explanations, no matter how counterintuitive, no matter how mystifying to the uninitiated, to everyone,
basically, no matter what, science is committed to giving a material explanation. So again, it's talked about on rare occasions. And again, some people say, dude, what's your problem? That's one of the basic rules of science. It always tries to find natural causes for things. But wait, given that it's an a priori assumption before you even pick up your calculator or your test tubes or whatever, what you've got here is any evidence you might discover is trumped by a philosophical assumption. So the evidence doesn't really matter, because we know before we even start that we're going to come up with some sort of material cause to explain what we see. So how does this cash out? Well, science looks at the world and collects data, but that data goes through a naturalistic filter, and the only thing that can come out the other side of that filter is a naturalistic interpretation, some physical cause, law or chance or something like that. In order to be scientists, this is what we have to do. Well, what comes out of that? Well, let's see, there can't be a historical Adam and Eve. Let's see, life had to get here somehow. So something like Darwinism, I guess, is true. And if our universe looks a little too fine-tuned, well, then I guess there's just billions and billions of other multiverses. Basically anything but God counts as an explanation as long as it's materialistic or naturalistic in that sense. So as a scientist, it's kind of embarrassing. This took me some time to learn, okay, because it's just sort of the water in which you swim. In the discipline I was in, we were mainly looking for naturalistic causes anyhow, but as I saw this as a broader picture, it's like, oh, this is really interesting. We're not really seeking truth, we're seeking just the best naturalistic explanation. And again, this revelation isn't new to me. It's been around for many years. C.S. Lewis makes a comment on this as well. Look, does the whole vast structure of modern naturalism, science is in its purview here, depend not on positive evidence, but simply on an a priori metaphysical prejudice? Was it devised not to get in facts, but to keep out God? It's basically what Lewontin was saying. Look, we have this a priori assumption that materialism is true, and just God cannot be allowed on the scene. So what intelligent design says is, wait. We look at the data, we study the world, but let's get rid of that naturalistic filter. Let's just look at what's there and say, hey, an agent's action might be possible. Can we detect it? Let's go see. And let's just go look out there and see for it. And okay, what would that look like? Well, we would see purpose in things. We'd see really sophisticated designs of things. We could look at something and go, how's that work? Can I reverse engineer it? Figure out what's going on? Again, can we see the design is the idea. And what I'm excited to see is that in a number of disciplines, people are taking up that approach. Biosystems engineering is one discipline that's basically saying something like this, like, get over that God thing. It's like really fruitful to look for the design and the purpose in these systems. And it's like, hey, it works. Look for design and you can see it. So to sort of wrap up this section, so you said law, chance, design. So when should you allow that design as a serious possibility? Well, easy. When it's very reasonable to do so. Okay, when is it reasonable to do so? When you're dealing with events that you can't take on your lab bench and repeat and repeat and mix and repeat and so on. You're dealing with non-repeating events like, where did we come from? Where did the universe come from? Where did the life come from? Those origin sort of things. I did ancient history. You see this in history as well, where we have to explain every historical event on purely human level terms. You can't have anything out there influencing the direction of history. Really? Well, what if something out there did influence the direction of history? Oh, yeah, well, we just don't see it. Yeah, good. Okay, so that's the problem. Oh, and then another area, because I did theology as well. It's in theology. It's hilarious. we have to explain away all the miracles in the Bible, right, as some misunderstood natural event. Really? Well, what? So you have to explain God away as a natural phenomena. Yeah, that happens in some theological circles as well. So in the process of being scientific as we do our theology. So my point here is this, that the goal of science should be to seek truth. That's what it says it's doing. So ask the average person on a college campus, what do scientists do? I'm not 6'5", but what do scientists do? It seeks truth. Yeah, do that, but without a philosophical bias is the point. So can we do that? And this is what intelligent design, it's sort of the lightning rod for advocating that. Let's study the universe, let's seek truth as we study the world without a philosophical bias of naturalism. So, again, that's some background to it just to help you with that. The fun part here is, okay, so I should consider design, right? So what's the best evidence for it? Good, I'm glad you asked, because we'll look at 2 out of 3 things here. One that I just sort of like for fun is to look at what the opponents to design arguments say. And then we'll focus the rest of our time here looking at biochemical systems, because they're sort of fun. And then, there's a whole series of talks that you won't get, where we look at the Earth and the universe, the fine-tuning in the Earth and the universe for evidence as well. But we'll just look at the biochemistry here, because that's IDE looks at all of these, but we've only got so much time, right? So anyhow, let me look at what opponents say. Francis Crick, the guy who discovered the structure of DNA and did a lot of the early theoretical work in terms of how DNA works and biochemistry, makes this comment. He says, look, biologists must constantly keep in mind that what they see was not designed, but rather evolved. Okay, so I'm looking through the microscope, and I go, oh man, it evolved. Yeah, okay. Oh wow, it evolved. Oh wow, it evolved. Okay, there's something that psychologists call that, but I forget what the term is. Okay, so why do I have to live in denial, right? I'm not in Egypt, so it's a problem. Okay, this is Richard Dawkins, the first sentence in his Blind Watchmaker, which is, look, biology is the study of complicated things that give the appearance of having been designed for purpose. Spends the next 300 pages of the book arguing that it's only the appearance of design. It's only the appearance. Hey, come on, it's only the appearance of design. Okay, well, again, why the denial? And even in the God delusion, his rave against, you can guess who, we live on a planet where we are surrounded by perhaps 10 million species, each one of which independently displays a powerful illusion of apparent design. Why do I have to call it a powerful illusion of apparent? Each one of them independently displays design. Come on, why do you have to deny this is the issue. And again, imagine that you're an art critic, and you go, okay, look, art critics must constantly keep in mind that what they see was not designed, but rather evolved. Or wait, no, we live on a planet where we're surrounded by perhaps 10 million works of art, each one of which independently displays a powerful illusion of apparent design. Okay, why do I have to be in denial? It's like, so to me, that's one aspect of, if you look at the way the critics argue, it's like, well, we'll grant apparent design, it's just not real. Well, let me show you some situations where I think it's reasonable to conclude it's real. So, little biochemistry background here. Proteins are made from about 20 amino acids, and they're strung together to make noodles, except each amino acid along the noodle is different. What amino acid is depends on the recipe that comes from the DNA. But again, just think of a protein as a noodle that folds up in a particular way. How it folds depends upon what amino acids are at what position, to give rise to very particular structures. Okay, so what kind of structures do you see from that? Well, let me show you one. This is a bacterial flagellum, and Michael Behe sort of made this popular some years ago as an example of irreducible complexity. This thing doesn't work unless it has all of its pieces put together, right? So what is it? Well, it's about 40 different proteins, and it's a rotary motor. It has a stator section, it has a rotor section, it has a drive shaft, it has 3 bearings in it. The hook region is, well, I'll have to show you the next slide to flesh this out a little bit better. There it is. Okay, that upper hook region is like a universal joint that, good, that works, that flexes so that the flagellum, the part that actually spins as the propeller, is able to have some flexibility. And notice the quote here that's fascinating. This comes from a secular journal, Cell, written by a secular author, just marveling over this molecular machine. He says, more so than other motors, the flagellum resembles a machine designed by a human. It's like, yeah, okay, that's kind of interesting, because if you've ever taken apart an electric motor or something like that, those are the parts that you see. There's the coil out there for the stationary part, there's the windings there and the rotor, and then it's got bearings that support it and hold it. Well, again, that's a nice picture of it, but let me show you in more detail what the proteins are doing to make this thing. A technique that's been developed in recent years is called cryo-electron microscopy, where they can visualize at the atomic level the structure of things inside of cells without having to crystallize it or do very, very fancy, sophisticated techniques. So what's pictured here, whoops, got to be careful what I press. Okay, what's pictured here is the basal body. So this is that hook region. These are the bearings, 1 bearing, 2 bearing, and the 3rd bearing is here. Oh, right there is the 3rd bearing, okay. There's a pump in it, that's the export apparatus. So down here is the part that pumps out the pieces that assemble to make the rest of the flagellum. It's kind of cool. This thing is assembled by ejecting its pieces out through a hollow part here in the shaft. But look at how those proteins have folded. Okay, that's what's kind of crazy with this. Those noodles, okay, have folded in these beautiful patterns to make extremely different physical structures here. And in the next slide, you get a cutaway view of it. Okay, this is kind of, so there's, again, the folded. Notice how flexible and open that thing looks because it flops, okay? And here's the bearings, here's the rod in the center. Here's the rotary part of the motor. The stationary part of the motor isn't shown. That's not part of the basal body. Here's the pump down here that pumps the proteins as it's assembling it up through that secretion channel all the way up. So there's about 40 different proteins that are assembled to build this thing. And just to give you another perspective on it here, this is again, a view of it where they rotate it, and it actually works. Yes, okay. Now notice here as it turns around, you'll see right down the hollow secretion channel, boop, right there. Okay, so again, the pump at the bottom pumps the pieces out that then are assembled to build the thing. And it's like, yeah, this just sort of happened by chance, just like this thing here just sort of happens by chance. It looks like a motor, doesn't it? All those noodles folded in just the right way to make this bacterial flagellum rotary motor that allows the bacteria to go and find food, is the basic function for it. So back to that journal Cell and that special article on molecular machines. This is a comment from the editor for that issue, where he says, look, we've always underestimated cells, and undoubtedly we still do today. But at least we're no longer as naive as when I was a graduate student in the 1960s. We now know that nearly every major process in a cell is carried out by assemblies of 10 or more protein molecules. And as it carries out its biological functions, each of these protein assemblies interacts with several other large complexes of proteins. Indeed, the entire cell can be viewed as a factory that contains an elaborate network of interlocking assembly lines, each of which is composed of large protein machines. Bacterial flagellum just being one example to show you. Well, why do we call these large protein assemblies that underlie cell function machines? Precisely because, like the machines invented by humans to deal efficiently with the macroscopic world, these protein assemblies contain highly coordinated moving parts. Well, who's writing this? Bruce Alberts, who was president of the American Academy for the Advancement of Science at the time. And again, he's the lead, he got all these articles together on all these different molecular machines, and he comments, "It's my hope that some of the young scientists who read this issue of Cell will come to the realization that much of the great future in biology lies in gaining a detailed understanding of the inner workings of the cell's many marvelous protein machines." Just don't say the D word or the G word with this, or you'll lose your job, and so on. But it's like, it's incredible. It's incredible, the complexity and the beauty of what we can see there. And to give you an example of it here, this is a little video that hopefully will play. Or did I stop it by mistake? Let me try it again.
Wouldn't it be amazing if there were a device that untangled knots automatically? Inside your body, there is. It's called a topoisomerase. It untangles knots in our DNA in an incredible process that keeps us all alive. DNA carries assembly instructions required for your body to generate new cells. But for DNA to do its work, it must be copied in a process known as replication. During replication, the DNA strands are separated, and protein machines use each strand to make a copy of the opposite strand. The end result is 2 copies of the original DNA. However, DNA replication faces a life or death challenge before it can complete its mission. Separating the DNA strands causes twisting in the portion of DNA that has not yet been separated. Eventually, the tension from the twisting increases so much that the uncopied segment of DNA wraps around itself in what are referred to as supercoils. If nothing intervenes, this buildup of supercoils will completely shut down the DNA replication process, since the 2 strands can no longer be separated, and your cells will die. Enter special proteins called topoisomerases to the rescue. Topoisomerases are amazing molecular machines that untangle knots. They come in 2 main types. Type 2 typically has 3 main sections: an upper gate, a middle gate, and a lower gate. Each gate can open or close during the protein's operation. A type 2 topoisomerase untangles DNA in 4 main steps. First, after 2 DNA segments enter through the top gate, it uses the middle gate to break one segment of DNA apart. Next, it passes the other DNA segment through this break. Then occurs the crucial step. The topoisomerase recombines the first DNA segment. The result is the elimination of 2 supercoils. Finally, now that the 2 DNA segments have been untangled, they are released by the topoisomerase. The second strand is released out the bottom, and the first strand is released out the top. Of course, this is just a simple summary. The actual process is a lot more complicated. Get ready for a more detailed version of how this process may take place based upon the latest research. When we zoom in on the upper part of the topoisomerase, one of the 2 overlapping DNA segments enters the upper gate. It then binds to the middle gate. Now the second DNA segment enters the upper gate. The DNA in the middle gate is cut in 2 by the topoisomerase. 2 ATP molecules attach to the upper gate, and the gate closes. One of the ATP molecules is broken apart into ADP and phosphate, releasing energy that may help keep the upper gate together during the next steps. The middle gate now opens and pulls the two halves of the broken DNA apart, creating a gap. Since the middle gate is connected to the broken ends, the DNA remains attached to it. The second DNA segment now moves through this gap. The middle gate closes and the attached DNA ends are reconnected. The upper gate rotates, which may prevent the second segment from moving back through the break. The remaining ATP molecule breaks apart, and the lower gate opens, allowing the second DNA segment to leave. Then the lower gate closes, the upper gate opens, and the first segment of DNA is finally released. The topoisomerase is now reset, ready to perform the same process all over again. The topoisomerase molecular machine is a wonder of the biological world. Its operation is highly orchestrated, displaying exquisite and intelligent design.
So again, that's the picture of this very, very simple molecular machine that solves what you saw there was a very, very difficult problem of the DNA tying itself in knots as part of the replication process. And I've just shown you two, but there's literally thousands of these different types of molecular machines in a cell that are, again, they act like machines. They're doing these mechanical processes, and again, it just sort of screams design is the point for it. Well, let me give you another example of one. This is sort of a classic example from skeptical websites. It's the backwards retina that vertebrates have. Our eyeballs are wired backwards. The light comes in there through the pupil of your, whoa. I gotta watch what buttons I press here, don't I? So there we go. There we go. The light comes through your pupil, the vitreous humor, and then here's the rods and cones in your eye that are actually detecting the light. But notice that the light has to go through a pile of nerves before it gets to the light detectors. So this is where skeptics have said, oh, the vertebrate eye is an example of bad design, right? Because your eyeball's looking backwards, right? The rods and cones should be up here on the front of the retina to catch the light directly rather than have it go through the nerves. And by the way, if you were an octopus, it would be wired correctly. The rods and the light detectors are here in front of the nerves on an octopus. So what, God got it messed up? You know, he did the octopus right and did you wrong? You know, so there obviously isn't a God behind this, right? Well, the response to this has been, well, wait. Detecting light is one of the most energy-intensive things that your body does. So the rods and cones need a lot of nourishment, which is provided by this pigmented epithelial tissue here, which has got a lot of capillaries and so on in it. So it's rather opaque, and the rods and cones need to be right next to that to get the nutrients that they need to detect light. If you're an octopus and you live in the dark, you don't need to have that level of nutrients on your light detecting cells. But vertebrates do. We do. So this is called, in engineering terms, constrained optimization. Given all the factors that you need to have, these things work best being wired backwards, if you will. So that, again, as I say, was the standard explanation that people would give for it until 2007. And I'm rotating the picture so that the next picture will make sense. 2007, some researchers studying the retina of guinea pigs, I believe it was, discovered that there's a particular cell in the nerve tissue there in the retina invertebrates. It's known as the Miller glial cell. Nobody quite knew what it did until they were doing their research. It expands like a goblet up here, and then next down to go through all that nerve tissue to get the light that was captured up here down to a specific cone. Each Miller glial cell is connected 95% of the time to a specific cone cell. And they looked at this and they said, oh, good grief, this is a fiber optic light guide. This is total internal reflection inside the Miller glial cells that keeps the light from scattering just in the nerve tissue, keeps the light contained in the cell until it gets down to the light-sensitive area. So this is like, compensating engineering need a lot of nutrients for those things. Need a lot of nutrients for those things, so they're on the backside. But you give each cell binoculars to get the light through the nerve tissue so that it isn't scattered on the way down. So they weren't not using the E word at all in the article that they're talking about this. So this is just incredible engineering, this is incredible design. Oh, by the way, they followed up on it with this, that the refractive index in the Miller glial cell is just such to totally internally reflect the red and the yellow and the green, but the blue light leaks out, and that's what the rod cells are sensitive to. So it's also a color filter in addition to a pair of binoculars for the cones. So once again, it's like this, what looked like bad design actually has this beautiful constrained optimization in terms of supplying the nutrients the rods and cones need. And oh yeah, let's give them binoculars as well to see through that scattering nerve tissue to give rise to clear images. So that's just like pretty incredible stuff. So let me give you another excursion off to another set of molecular machines and another fun issue in the world of biochemistry. This involves, again, the DNA is where your recipes are stored. They're photocopied into RNA inside the nucleus, and then the RNA goes out of the nucleus into the cytoplasm, where where machines called ribosomes do the hard work of reading that recipe, if you will. Take the RNA and then build the protein from it. So I'll give you a cartoon here that shows that a little bit bigger. So the recipe to make the proteins in your body in most organisms is written in a 3-letter code, a 3-nucleotide code that is kind of illustrated here. So let's see, there it is. Okay, so groups of 3 nucleotides on the RNA are read within the ribosome and, again, give rise to what amino acid is put in at that position in the growing polypeptide chain, in the growing noodle, if you will, for it. So one of the mysteries was, okay, what is that code on which these protein recipes are written? And just to give you a little picture of what the ribosome is, it's a very simple molecular machine in and of its own right. But the code, when this was figured out, the guys got a Nobel Prize for it. So there's 4 possible nucleotides, so that's why there's 4 times 4 times 4 in terms of the letters that you see there. And the recipe, basically, the reading of the recipe is, UUU or UUC, put phenylalanine in the amino acid at that point. Any of those 6 would put leucine in the growing polypeptide chain at that point. These 3 would put isoleucine there at that point. And again, what this basically is a conversion table, right? Here's the recipe, and then this is the reader, the decoder ring, the language. There's several ways to think of this in terms of, okay, how does the recipe get turned into a particular protein, a particular amino acid, and thus a particular recipe makes a particular protein. Well, Francis Crick, the guy I mentioned earlier, who said, you always got to remember that these things just evolved, predicted that a code like this would exist in the DNA, and said that, when we discover it's there, we will realize that it's a frozen accident. Why an accident? Well, because life just got here by chance, right? So in some warm little pond billions of years ago, something got together, and this is the decoder ring that it decided to use, right? And it's got to be frozen because once you have your code, you can't change it because you get garbage out the other end, right? The recipe that with this code makes, functioning proteins, you change the decoder ring and you get garbage out of it. So for the thing, it shows up by accident and then it just has to be frozen. And this was one of the strongest arguments you would get in a biology class for common ancestry, that everybody has it. They're virtually the same thing, from viruses and bacteria, up to us. We all basically have the same decoder ring. How did that happen? well, it must be that we all come from a common ancestor, that first frozen accident cell that we've all come from. So again, big argument from the naturalist side. Well, some people asked a question of this. There's a lot of redundancy in this code, as you notice. 6 things there that can make leucine, right, and so on. So they said, I wonder how error tolerant this code is. In other words, if a mutation occurs in the DNA, how drastically different is the amino acid that gets put in? I mean, instead of being an acid, is it a base, or is it hydrophobic, or something like that? How different is the amino acid that's put in if a mutation occurs back here in the recipe? So they wrestled with that. It's kind of a theoretical question to deal with in the world of biology. But they basically said this, is like, okay, given the redundancy that's there, theoretically there's about 10 to the 18th genetic codes that have that same level of redundancy, reducing 4 times 4 times 4 nucleotides down to 20 amino acids plus 3 stop codes or so. And then this is this is a feel for how damaging that code would be to the protein that code would make. Okay, so like I said, it's a bit theoretical. But here was the issue that was shocking. What you would expect if our code was a frozen accident is that it would be like up here someplace. That's where the most of the codes fall out in terms of how damaging they are in terms of mutations. But our code is there. It's like, whoa, way off on the side. Now, because of all the theoretical assumptions, you can't quite say that we know we've got the best possible genetic code in terms of error tolerance, but it's a real good one, okay? That much you can say. And people have gone on and tried to say, well, could this code have evolved? And again, the latest work on this is, in the quote there, that no, there's no imaginable, simple evolutionary pattern that can allow the first life to sort of experiment and find a code like this that works. It's like, no, this thing really does have to pretty much be a frozen accident, it seems like. Well, Hubert Yockey has wrestled with this and said, look, 10 to the 18th is a little bit too generous. If you really ask what are the total possible number of codes that you're dealing with, it's more like 1 times 10 to the 70th possible codes out of, again, 3 nucleotides, generating something. You've got about 200 million years to get life started on the Earth. So in that warm little pond that's trying to find a decent genetic code, it's got to be testing 10 to the 55th codes per second to find one that works. That's not very likely, okay, is this point. Manfred Eigen, who is a pretty flaming atheist in his day, he's no longer with us, so I hope his theology has changed, but he was with Crick in this sense of, wait, it's got to be a frozen accident, that this had to be there with the very first cell, and because you can't change the decoder ring, right? You just get garbage out, which would be death. So the code has to be there in the very first life. Well, the dilemma here is, and let me just give you a quote where Simon Conway Morris, where he's more favorable, is like, well, wait, no, this thing really shows, again, eerie perfection, starting evidence of optimization. So instead of this thing being a frozen accident, we're all common, and there's some common ancestor to all of us, this code is there because it has a function. It minimizes the effect of mutations in the DNA on the proteins that result. If the code has a function, you can't call it an accident. And why would it be the same or largely the same in most organisms? Because it works well. So what was a very strong argument, right? Frozen accident, we all have it, so therefore there's a common ancestor. It's like, well, wait a second. This thing serves a function. There's a reason for it to be there, and that we have so many organisms all have the same code. So one of the things I see in the biochemistry world is basically this, that people might call it junk. People might say, oh, it's a frozen accident or something like that. But as we better understand what's going on and how the thing works, you discover design popping out of that. The better we understand it, the more design shows up. So that makes biochemistry actually kind of fun, because it's like, oh wow, gee, we're seeing God's hand more and more in this. So again, I think the problem here is that for Darwin, and again in the 1800s, people thought that life was just chemistry. Get some acids and bases and nucleotides together, and that's what's in the cytoplasm and so on. So life is just chemistry, But then we started to understand things a little bit better, and life got a little more complicated. And when we discovered enzymes, those noodles, and how they fold into really complicated structures that give rise to things, well, I guess life is complicated chemistry. Okay. So, and then we go on and we discover DNA and how DNA works and interacts with proteins and RNA and so on. And class, I think life is just really complicated chemistry, okay? And then we went on and we discovered these molecular machines, okay? ATP synthetase is one fun example of this. This is in your mitochondria, this is in plants, this is the thing that makes the gasoline that powers your cells, okay, ATP. And it's a little rotary motor. You can see the thing spinning. So it's like, wow, there's these molecular machines, these motors and so on going on inside of cells. It's like, life is just like really, really complicated chemistry, And then, then this little molecular machine that makes the proteins and the other molecular machines that are around those ribosomes and so on. Those are just like, look, life is just really, really, really complicated chemistry. This is the stage that I got when I was taking biochemistry myself. Life is just like really, really, really complicated chemistry. Then you start mutating the individual amino acids in the ribosome, and it breaks it. And it breaks it. And that breaks it too. It's like, man, these things are just highly, highly optimized machines as well. I mean, look, life is just like really, 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 really complicated chemistry. And then, fortunately, some guys from another building came wandering by one day and, hey, what you doing? Oh, yeah, let's look at, hey, we do stuff like that too. Except we put it on server racks and so on. And it's like, I think you guys have the wrong metaphor for life. Life ain't chemistry. Life is actually information processing. It's a whole different league than what people had ever imagined. So again, the cytoplasm of, cells are just this little bags of garbage, this protein garbage inside. It's like, no, I'm sorry. There's some pretty sophisticated microprocessors going on inside of there. And it's like, you have to think of cells as information processing systems. And you got to think of these things as stuff that would put Intel out of business in 30 seconds. I mean, the incredible hardware, incredible software, operating systems, firmware, and they reproduce themselves as well. It's like, these things, you have to see them as computers in terms of the level of complexity of what's going on inside of them. And just to give you one example of this is a purple sea urchin. And the person who's pictured there is Eric Davidson. He was at Caltech. He spent his life studying the embryonic development of purple sea urchins. Why would you spend your life studying purple sea urchins? Well, I just by chance, I think God had his hand in it, I heard him give a talk on this many, many years ago, early in his career and mine, where he said, look, why purple sea urchins? Because I just go down to the beach, look around in tide pools, and I can grab a bunch of these and fill a bucket and bring it back to the lab. And these things are just egg and sperm factories, okay? They just blow out sperm, they blow out eggs. It fertilizes in the open water. So I can just, have my buckets of purple sea urchin, take the eggs, take the sperm, mix it in a petri dish, the eggs fertilize, and then I can watch them develop them from scratch. It's like, and PETA doesn't care. So this is awesome, okay? We've got this baby. So, like I said, he spent his career watching these things develop from fertilized eggs, splits in two, splits in two, splits, and watching these things develop. And he also developed dyes and fluorescent markers and other things that could tell him what particular gene was turned on in what particular cell. Again, he had an army of grad students and an army of postdocs that worked with him on this. And this is just one example. So the red cells there are the micromere. It's something that shows up later on in the embryo. And in the micromere, the gene PMR1 is expressed. And if PMR1 is expressed, it blocks the gene R of MYC from doing something when the gene product UB shows up. So in the micromere, because R of MYC is not expressed, when UB shows up, ALEX1, delta, TBR, and ETS1 show up, are expressed, and do their thing in those cells. In all the other cells in the embryo, the clear ones there, PMR1 is not expressed. So when UB shows up, R of MYC is expressed. Oh, sorry, yeah, okay. Yeah, when UB shows up, R of MYC is expressed, which then blocks ALEX1, TBR, delta, and ETS1 from doing anything. So again, he spent his career looking at how these things, what genes are on, how they interact with each other, and he's studying what's referred to as these developmental gene regulatory networks. I won't take you through these slides, but you get interactions between cells and between tissue types in the developing embryo as well as this goes on. But I will show you this one. This I got from his website, and unfortunately you probably can't read it, but this is the endomesoderm tissue from 21 to 30 hours after fertilization. Okay, what tissues are showing up and what genes are expressed and how they're interacting with each other and turning some on and turning other ones off. And electrical engineers look at this and go, that's my job. I design stuff like this. I do not design stuff like this by shooting a shotgun at a piece of silicon, That's like design. I'm sorry, It's like, whatever. Well, let me just carry this on and show you one of the genes, Endo16, just to flesh this out, because I found another article that his group had published. Okay, the Endo16 gene has a promoter region. That's what tells it when to turn on, when to turn off, how much to turn on, and so on. The promoter region that helps modulate the development of the endoderm, whatever the endoderm is. It contains, this Endo16 gene has 34 binding sites for 13 different transcription factors and cofactors, and they break it into 6 modules, okay? And what the purpose of the article was to focus on modules A and B and to try to figure out the internal logical wiring of what binds, what's binding, and does that turn it on or block it or turn it off, or how much does it turn it on? So again, he spent his career working on the circuit diagram for how that purple sea urchin embryo goes from a single cell up to the adult phase as far as he could follow it. So what's Eric Davidson say about it? He says, look, what emerges is almost astonishing, almost. Okay, a network of logic interactions programmed into the DNA sequence that amounts essentially to a hardwired biological computational device. Okay, so these body plans, and that's what he's looking at here, right? These gene regulatory networks are what makes a sea urchin, or a squirrel, or you. It's like that's what he's studying. These body plans are created by the logic outputs of network subcircuits. And in modern animals, these outputs are impervious to continuous adaptive variation. Boop. Wait. Charles Darwin argued for numerous successive slight modifications. And he just said, wait, what makes an animal, its body plan, is impervious to numerous successive slight modifications, unlike genes operating more peripherally in the network. So what's Davidson just said? Darwinism cannot explain where these body plans came from. That's what's wild. Okay, then he said it even more explicitly here. Neo-Darwinian evolution is uniformitarian in that it assumes that all process works the same way, so that evolution of enzymes or flower colors can be used as current proxies for study of evolution of the body plan. It erroneously assumes that change in protein coding sequence is the basic cause of change in developmental program. It erroneously assumes that evolutionary change in body plan morphology occurs by a continuous process. All these assumptions are basically wrong. It looks designed. I'm sorry, that would be the conclusion I would draw. And he says, look, this cannot be surprising, since the neo-Darwinian synthesis from which these ideas stem was a pre-molecular biology concoction focused on population genetics and adaptive natural history, neither of which have any direct mechanistic import for the genomic regulatory systems that drive embryonic development of the body plan. So basically what Davidson is saying is, yeah, Darwin works to explain flower colors and little tiny changes, microevolutionary changes just fine. But what Darwin was hoping to explain, claimed he was able to explain, was where the body plans came from. How do new body plans show up in the world? And now that we understand how these gene regulatory networks work, how complex they are, how interactive they are, we got to be looking someplace else than for Darwinistic mechanisms to explain it. So again, just to give you further background on this, this is what Steve Myers spends his time on in the book Darwin's Doubt, saying, look, our best science is telling us that this stuff looks designed. If it walks like a duck, if it quacks like a duck, if it looks like a duck, maybe it is a duck, okay? And we should take off those naturalistic blinders and recognize we're dealing with stuff here that has no simple way it could have gotten here by law and chance, that we're looking at things that are designed. So as a Christian, how I would summarize it is the way it's done in Psalm 139. For you created my inmost being. You knit me together in my mother's womb, and I praise you because we are just fearfully and wonderfully made. And the more we study, the more incredible God's wonderful works seem to be. So that to me is what's so impressive with it. And one final thought, and what I find interesting about this, is that as humans, we first started to understand computers and invent and develop software and computer codes and so on, 1940s, 1950s, and so on, just about the time that Watson and Crick are deciphering DNA, and Crick is proposing that there's codes in that DNA that have various functions. There's that decoder ring and so on that are there. That really the hallmark of life isn't chemistry. The hallmark of life is information processing. And I just kind of look at that parallel development of the more we've come to understand computers, now we can better appreciate what's going on inside of cells, inside of ourselves, inside of life. And to me, it's basically God saying, dudes, you've done a pretty good job with silicon-based information processing. Now you can better appreciate the carbon-based processing that I've done, right? It's like, yes, okay. So again, to wrap up, intelligent design, well, I've tried to show examples and so on where it's like the design is just kind of screaming that it's obvious. And again, my point is that science needs the freedom to go beyond just law and chance as the explanations for that, and to follow the evidence where it leads. And again, if it looks designed, maybe it is designed, is our best answer for it. But there's a problem with that. And it's more than just, science only focuses on naturalistic explanations. The problem is that a design conclusion, the universe is a little too darned orderly to be just a big accident, is scary. Quite frankly, it's scary. And this is where I love being a Christian, because I can say, dude, it's designed, and that is scary. But I have a suggestion about who the designer is that I can back with evidence from other areas of apologetics just to flesh that out. But the designer that I have in mind is one who we don't need to fear, because if you put that amount of effort into making us, maybe it's because he loves us. And not only does he love us, but he's also come to rescue us and to redeem us. So we can recognize design and look beyond the scariness of a designer and embrace him because he's come to rescue us and he loves us. So the full story ends up with, again, a happy ending, that if you follow the evidence where it leads, you get the design, and you can get then to the designer, who I think are many, many good arguments that it's Jesus, who's our creator, and he actually sacrificed himself to rescue us. So let's close in prayer here then. So, Father, we just thank you for the tremendous progress people have made in understanding your world, and from telescopes to microscopes to cryo-electron microscopy, we can now see your handiwork at so many levels. I just pray that you would use that evidence to soften people's hearts, to draw them to you, to recognize there's got to be something more than just material in the universe, and that you might use Christians as your ambassadors to draw people to you. And just pray this talk might work to that direction as well. We just ask you to bless us to overcome the cultural stigma that has been in so many quarters, with the evidence that you are who you say you are, and people can trust you and be redeemed. So again, we ask for your blessing now. In Jesus' name, amen.